The bed bugs.
Ugh.
Just saying the words can make you feel a bit itchy, can't it?
Oh, absolutely.
These tiny things, they are almost, a thing of the past after we used DDT everywhere.
Mm-hmm. Decades ago.
But now,
Yeah.
they've properly roared back. And the big question is how? How are they managing to just shrug off everything we throw at them? That's what we're digging into today.
And what's really fascinating is that scientists, researchers at Virginia Tech, they're giving us some pretty solid answers.
Yeah.
Yeah, they've found not just one, but actually two really key pieces of the puzzle. It's quite something.
Okay, tell me more.
Well, first, they've pinpointed a specific genetic change, a mutation, that seems to be helping the bed bugs resist pesticides.
Ah, the resistance factor. Okay.
Exactly. And second, and this is a big one, they've actually mapped out the entire genetic code, the whole genome of the common bed bug. First time ever.
Wow. Okay, so this is like getting the inside scoop direct from the researchers who did the work.
Pretty much.
We're going to explore how these breakthroughs are, well, helping us understand these little pests better. And maybe, just maybe, point towards some actual solutions down the line.
It's a classic story, really, when you think about it. We nearly wiped them out with DDT back, in the 50s?
Yeah, post-war era.
Right. Then DDT gets banned for good reasons, environmental concerns, health issues.
Of course.
And bam, slowly but surely, the bed bugs start to reappear. But it wasn't just reappearing, was it?
No, it sounds like they came back. Different, tougher.
That's it. They evolved. They developed resistance to the newer insecticides we started using. Makes controlling them incredibly difficult now.
Like they're always playing catch-up, but they're faster.
Yeah, something like that.
And that leads us right into that first big discovery you mentioned from Virginia Tech, this specific gene mutation.
Mm-hmm. This was work led by an urban entomologist there, Warren Booth, published in the Journal of Medical Entomology.
And how they found it is, well, it's kind of a cool story in itself, isn't it?
It really is. Professor Booth apparently called it a bit of a fishing expedition.
A fishing expedition. Why?
Well, the main goal initially wasn't just finding a resistance gene. It was partly, a project to help one of his graduate students, Camille Bloch, get her feet wet with molecular biology techniques.
Ah, okay. So a training exercise that hit the jackpot.
Sort of, but, Professor Booth, he works on urban pests, so he probably suspected resistance was linked to genes like we see in, say, cockroaches or whiteflies.
Right, makes sense.
Okay.
So he likely guided Camille towards analyzing DNA from lots of different bedbug groups.
Exactly. And they looked at a lot of samples, DNA from 134 different bedbug populations.
134. Wow. Where did they get them all?
Pest control companies sent them in, collected right across North America over like a 14-year period, 2008 to 2022.
Okay.
And Camille, she analyzed generally one bug from each of those populations.
And the timing on this discovery was apparently really something.
Oh, yeah, get this. She found the mutation in her very last set of samples, like the final 24 she analyzed.
No way. Talk about down to the wire. Especially since you said she was quite new to this kind of lab work.
Exactly. Imagine that moment. It really shows how, learning and discovery go hand in hand in science. Developing those skills was key, and boom, a major finding comes out of it.
And why was testing just one bug per population usually enough?
Well, often with bedbug infestations, there's not a huge amount of genetic diversity. Lots of inbreeding happens.
Oh, okay. So genetically quite similar within one group.
Generally, yeah. But they didn't just stop there. Once they found the mutation in those two populations...
They double-checked.
Oh, absolutely. They went back, tested all the available bugs from those two specific groups, and guess what?
Every single one had it.
Every single one. Confirmed it wasn't just a fluke.
Okay, so what's special about this particular mutation? What gene is it in?
It's in a gene called RDL. And the interesting thing is, in other insects, this same RDL gene is known to make them resistant to an old insecticide called dieldrin.
Dieldrin. Haven't heard that name in a while. That's just been banned for ages, right? Since the 90s?
Correct. But here's the kicker. Dieldrin works in the same way. It targets the same part of the insect nervous system as a pesticide we do use a lot today, Fipronil.
Fipronil. Wait, isn't that it? Like flea and tick stuff for pets?
Bingo. Exactly. For dogs and cats.
Okay, so how could those be connected? The pet stuff and the bedbug resistance?
Well, the thinking, Professor Booth's hypothesis, goes something like this. Both dieldrin and fipronil mess with the same target in the bug's nerves.
Uh-huh.
So if a mutation changes that target slightly, making dieldrin less effective, it might well do the same for fipronil.
Right.
So imagine this. You treat your pet with fipronil, Maybe some bed bugs lurking in the house get exposed to low levels, perhaps through shared bedding or just being nearby.
Okay.
It might not be enough to kill them all outright, but if some bugs happen to have this WWII mutation, they might survive better than the others.
Ah, so the fipronil use could unintentionally be selecting for this resistance mutation in the bedbug population.
That's the idea. It gives the mutated bugs a survival advantage, they reproduce, and pass the resistance on.
Huh.
Okay.
That's actually quite concerning when you think about the ripple effects. But I guess there's still unknowns here.
Oh, definitely. Big questions remain. Like, is this mutation brand new, or has it been kicking around in bedbug populations for a long time? Maybe at low levels, we just don't know yet.
And finding that out is important, right? For understanding how they adapt.
Crucial. Yeah. So the next step for the team is to broaden the search. They want to look for this mutation in bed bugs from other places, like Europe.
Okay.
And also, and this is cool, they want to look at DNA from really old bed bug specimens, the ones kept in museum collections.
Go back in time genetically. That could tell us when this resistance really emerged.
Exactly. Give us a historical timeline.
Okay, so that's a massive step in figuring out how they're resisting. But you said there was another major achievement from the Virginia Tech Lab.
Yes. Hot off the press, basically. In November 2024, Booth's lab managed to sequence the entire genome of the common bed bug.
The whole genetic blueprint.
The whole thing. Published in the Journal of Heredity. And like I said, this is the first time anyone's done it completely. It's a landmark achievement.
Wow. So what does having the full genome actually allow researchers to do? What are the possibilities?
So many things. Professor Booth pointed out one big advantage. Analyzing that old, often damaged DNA from the museum specimens we just talked about.
How does the full genome help with degraded DNA? I don't quite get the link.
Okay, think of it like having the complete instruction manual for building something really complex, like a spaceship.
Okay.
Now imagine you only find a few torn, faded pages of those instructions from an old, wrecked spaceship. If you also have the complete, pristine manual as a reference, you can figure out where the torn bits fit. And maybe even reconstruct the missing parts based on the overall plan. Exactly. The sequenced genome acts as that complete reference, a chromosome-level template. Researchers can take those fragmented bits of old DNA, align them against the complete genome, and piece together whole genes, maybe even bigger chunks, from those historical samples much more accurately.
Okay, that makes perfect sense.
Yeah.
So it's not just about history. This genome map has practical uses now. For pest control, maybe?
Absolutely. The Booth Lab works with pest control companies, remember? Understanding the full genome could help track how different bedbug populations are spreading globally.
Right.
It could reveal genetic variations linked to different behaviors or maybe different responses to treatments. All that information can feed back into making eradication strategies smarter and, hopefully, more effective.
It really shows how that deep fundamental science can lead to real-world applications.
Definitely.
And it sounds like the graduate student, Camille Bloch, she's really found her calling in this area, urban evolution.
Yeah. Reports say she's really enthusiastic about continuing this kind of work. And she made a great point, bed bugs are something a lot of people can unfortunately relate to.
Too true. Many of us have either had encounters or know someone who has. It makes the science feel more immediate somehow.
It does. That personal connection, even if it's an unpleasant one, makes people pay attention to these kinds of discoveries.
So pulling these two big advances together, finding that specific resistance mutation and mapping the entire genome, we're really getting a much clearer picture of these persistent little critters.
We are. We're unlocking their genetic secrets. Both the specific tricks they use to survive our attacks, like the WWII mutation, and their entire genetic makeup. It's building a much stronger knowledge base.
And that knowledge is key, presumably, for fighting back more effectively.
Precisely. This isn't just science for science's sake. It's laying the groundwork for developing, more targeted, maybe smarter, and ultimately better ways to control them in the future.
Okay, so wrapping up this deep dive, it leaves us with a pretty important thought, doesn't it? Now that we have these powerful genetic insights, what kinds of new strategies might emerge? Could we see innovative, maybe more environmentally friendly ways to finally get the upper hand on bedbugs?
That's the multi-million-dollar question, isn't it? It's certainly going to be a fascinating area to watch going forward.
Definitely something for you to think about.